about some experiments looking at the regenerative capacity of the pig heart. Thank you.
Yeah, thanks for introduction. First, I would like to thank Jay and James for invitation and joining this wonderful symposium, and also giving the opportunity to share my research here. So my group and also Jay's group all studied the porcine heart regeneration capacity. So here I will show the results in my group. As we know that the zebrafish and the newts can regenerate cardiac tissue throughout life, and mostly achieved by cardiomyocyte proliferation. However, adult mammalian heart does not have the ability to replace functional myocardium, which was lost after myocardial infarction. And the growth of mammalian heart after birth is majorly achieved by hypertrophy of pre-existing cardiomyocytes. And recently, it is established that neonate mice can regenerate cardiac tissue after injury within 7 days after birth. And this regeneration is achieved by proliferation of pre-existing cardiac mice after injury. However, it's unknown if the hearts of young, larger mammals can regenerate after injury, especially myocardial infarction. So the aim of this study is to investigate whether the hearts of larger mammalian animals have capacity to regenerate after injury, and when this is switched off after birth. So in this experiment, we used piglets, which was on day 2 after birth, and day 3 and day 14 undergo open chest sham surgery or have coronary artery pulmonary ligation. Cardiac MRI imaging was performed at week 1 and 12 weeks to see the imaging, the scar tissue, and also determine the heart function. And also echocardiographic function was performed at 2, 4, 8 weeks, and 12 weeks to follow up the changing profile of the ejection fraction. And some piglet was euthanized at week 1, at week 4 to explant tissue, and the remaining pig heart was explanted at 12 weeks to undergo immunohistology studies. And the BRDU was injected to trace any dividing cardiomyocyte on days 0, 3, and 6 after surgery. And also we performed the RNA-seq using the pig heart tissue on day 1, day 7 after surgery. To ensure the successful creation of the myocardial infarction, all the animals received the myocardial injury will have ECG monitored to ensure that ST segment was elevated. This was further confirmed by monitoring the cardiotropin I in blood samples at baseline and 24 hours after infarction. As we see here, 24 hours after myocardial infarction, the cardiotropin I in the blood was elevated about 50%, and no significance was found across all 3 groups, indicating the severity of the injury was similar in all 3 animal groups. Echocardiographic functions showed that piglets in group 1 had better and early cardiac function recovery after MI. As shown here, all the animals in sham groups have similar ejection fraction from 60 to 65%. However, after injury, all animals have significantly reduced ejection fraction as you here. But in group 1 animals after MI, they have better recovered the ejection fraction as compared with group 3. And maximal recovery of the ejection fraction was seen in group 1 at 4 weeks, which was significantly higher compared with group 2 and group 3. And this ejection fraction was maintained until week 12. And a delayed recovery of the ejection fraction was found in group 2 starting from week 8. Now, however, in group 3 after MI, the ejection fraction was significantly lower, even up to 12 weeks. Using cardiac MRI imaging, we quantified the ejection fraction. We found at week 1, all animals in group 1 to 3 have reduced ejection fraction to 60% of the sham group. However, only in the group 1, they have significantly recovered the ejection fraction, which was about 90% of their respective sham at 12 weeks. This panel shows the MRI imaging of the injury. The arrow shows where the injury site. We can see some defects here indicate the myocardial infarction. At 12 weeks, however, we did not observe the significant defects in the anterior wall of the group 1 piglets. On the contrary, we saw significant defects, perfusion defects in the group 3 animals at 12 weeks, indicating scar formation in these animal groups. We quantified the scar area, showing that at week 1, across all 3 animal groups, the scar area was about 15 to 18%. No significance was found in this at 1 week. However, at 12 weeks, the scar area was significantly reduced from 15% to almost 6%, which was significantly smaller compared with group 1, group 2, and group 3 animals. So then we harvested tissue pig heart to perform Mason triclosanine to visualize the scar formation. We found actually minimal scar tissue was found at the injury site. However, in group 3, mature and transmural scar was formed, indicating group 1 had some repair mechanism. So we quantified the wall thickness as shown here. All 3 groups has reduced the wall thickness at injury site, but the reduction in group 1 was less as compared with group 3, and the fibrotic tissue area at injury site was minimal and was significantly less compared with group 2 and group 3. As we inject the BRDO, we perform the BRDO immunostaining to look at the BRDO incorporation into the cardiac cells. As shown here, we found extensive BRDO incorporation into the myocardium, and a lot of the cardiomyocytes incorporate BRDO into their nucleus. We further look at the 1 week and the 4 week post-MI to see the BRDO incorporation in group 1 and group 3 at proximal and the distal border zone of infarct. We found quite a lot BRDO incorporated nuclear in the group 1. This was significantly reduced in group 3. Quantification of the group 1 BRDO positive cardiomyocyte showed that at week 1, at the border zone of infarct, the rate of BRDO positive cardiomyocyte was similar to sham. However, at week 4, the rate of the BRDO cardiomyocyte significantly increased to 7% or 12% at the proximal or distal border zone. Similar pattern was found in the group 3. However, the rate of BRDO cardiomyocyte was much less compared with group 1, indicating group 1 have more actively DNA synthesized at week 4. So next we look at the cardiocytokinesis. We defined cardiomyocyte cytokinesis as they must express aurora B protein with disassembling sarcomere and a cleavage furrow. As shown here, the picture shows in the group 1 and the group 3, we both found the dividing cardiomyocyte. The rate of the aurora B expression cardiomyocyte was similar at week 1 and week 4 in sham-operated surgery animals. However, myocardial infarction stimulated a significant increase in the cardiomyocyte dividing, which was maintained until week 4 and then significantly dropped down at 12 weeks. We found, interestingly, that the myocardial infarction did not increase the myocyte dividing rate, and which was significantly dropped down at week 4. So the big difference was found at week 1 and week 4. So indicating some things, cardiomyocyte dividing is happening from week 1 until week 4 in the group 1 animal heart. We also performed the Western blot to quantify the aurora B protein expression. We found actually in group 1, aurora B protein expression was significantly upregulated as compared to sham and also group 3, indicating active cell cycle is happening in the myocardium of day 2 piglets. And we also looked at MASP1 protein expression. However, MASP1 protein expression in the myocardium infarct pig heart was similar compared with sham, or even less in myocardial infarct group, in group 3, and compared with their own sham. So indicating that progenitor contribution to dividing cardiomyocyte is minimal. We also performed RNA-seq analysis to determine the gene expression levels. As shown here, we found more than 300 genes involved in cell cycle was upregulated in group 1 animal on day 7 post-MI compared with group 3. On day 1 post-myocardial infarction, we found significantly upregulated genes involved in myocardial fibrosis was upregulated in group 3 in the infarction area and also border zone of infarction, which was significantly higher compared with group 1 animals. Similarly, on day 7, the same pattern was found. We found even more magnified gene expression of fibrosis in group 3 at infarct zone and the border zone of infarct, which compared with group 1 animal, indicating fibrosis is happening in the group 3 animals, while some cell cycle or cell kinesis is happening in the group 1 animals. So this data support that cardiac injury caused by cardiac infarction can be largely repaired in the hearts of day 2 old piglets with minimal scar in injured tissue, and the repair mechanism is lost very soon after day 2. The cardiac repair mechanism is characterized by a robust increase in cardiomyocyte regeneration. So these studies was carried out in my lab, and with RNA-seq study performed by Stuke from Duke and also with collaboration with Jiezhang. And this study was funded by the National Medical Research Council of Singapore and also supported by the Sinhealth Research Foundation. Thanks for your attention.
Thank you, Lee. Got questions? Reza?
Congratulations, this was a great study. The BRDU experiments that you did focus mostly on cardiomyocyte proliferation, but the mechanism for cardiac repair in addition to cardiomyocyte proliferation requires resolution of fibrosis. Did you look at proliferation or activation of fibroblasts in the 3 groups to see how they play a role in cardiac regeneration?
Yeah, I think good suggestion. Currently, I did not look at the fibrosis, but definitely extracellular measures of fibrosis, whatever signaling pathways, I believe definitely will involved in this regeneration process. But really how they behave, how to contribute this one is unknown. But based on the RNA-seq results, we actually know they have turnover between day 7 and day 14. You can see the fibrosis gene, a lot of fibrosis was upregulated, but fibrosis gene in the day 2 piglets was actually inhibited. So I believe, yes, but currently we are not looking at this side. Yes,
It's great we now know that larger mammals have a regenerative ability as well, but I'm just wondering if you have any insights to the degrees. Are they roughly similar, mouse to pigs, and can we expect humans to be similar?
Yes, true. Actually, when I was in Jazz Lab, we also did some preliminary studies. So we hypothesized at that time because the pig, the lifespan is more than 10 years. We expect the window for regeneration may be even longer compared with rodent, maybe 14 days. So actually, I started the first experiment with day 14. Then actually, we only found the mature scar was formed. Then I go back to day by day until day 2. Actually, we found this. So in refer to the human, I predict maybe even short regeneration window in human.
The pig myocyte is very special. They can have up to 16 nuclei.
Yeah.
So multiple nucleation happens in the first few weeks. That may contribute to the remuscularization and early in P1, P2 animals also, but we do not know.
Yes. Yeah, sure.
Okay, thank you very
Thank
much,
you.
Lee.